Hello and welcome back to our next lecture. Today we will talk about genetic variation. Let's just recap what we talked about previously. So in the last lecture we talked about meiosis. And meiosis is, of course, the production in the end of gametes. And gametes, when they fuse, produce the next generation. So we will talk in the next few lectures about genetics and genetic heritability and the passing down of traits from one generation to the next generation. And of course, all that has to do with DNA, and we talked about DNA at the very beginning of the course. And so today, we're going to spend a little bit of time thinking about meiosis, thinking about DNA, thinking about structure of DNA, and then thinking about what kind of variability can occur when changes in DNA occur. And they can happen for a variety of reasons. And then the most basic one is meiosis. So we talked about meiosis last time. We said that in meiosis, but not in mitosis, the homologous chromosomes are separated. So the homologous chromosomes are the parental chromosomes, the chromosomes that come from the mother or the father. In meiosis, paired homologs exchange chromosome arms, and that is called crossing over. So that is where some variation can occur. And this shuffles parental genes and creates variation between offspring and parents. We said that meiosis 2 is quite similar to mitosis, and that during meiosis 1, the homologous chromosomes are separated. Then we said that during meiosis 1, chromosomes can align in the metaphase plate in two different ways. We had the left or right of the equator example, where you have the two red ones on the left and the two blue ones on the right, or you have one left that's blue and one left that's red. And so therefore you have the variability just by the way that the chromosomes, without any crossing over, can align in different ways and can therefore create some variability. So that leads to different possibilities of combinations in the finished sex cells. And then we talked a little bit about cell cycle, and we talked about the regulation of the cell cycle, how CDKs and cyclins are involved in that. And we talked about genes that have to do with promoting or suppressing the cell cycle in general, and how that when they go awry, when something goes awry with them, how that can lead to cancer. So today we want to talk about DNA changes, and we want to see how changes in the DNA can be both advantageous or disadvantageous, and then how you can use some biotechnology to look at variation between individuals. All right, so let's take a look back at DNA structure. So way back at the beginning of the semester, we talked about DNA and RNA and protein. We talked about the fact that there's transcription that occurs to produce RNA, that there's translation that occurs to produce proteins. We talked about the code and said that 3 nucleotides code for one amino acid, and that the different types of amino acids, whether they're polar or whether they are non-polar, maybe they're even charged, that can have an effect on the formation of the 3-dimensional structure of proteins. So we looked at that table back then, and we said, well, if you have a change in your codon, then that means that the change in the codon also, in some cases, but not all, leads to a change in the amino acid that is used in the protein. And so what we have here as an example, when we want to talk about variation today, is hemoglobin. So this is hemoglobin. That's a protein that is part of blood cells, carries oxygen. And when hemoglobin is altered in certain ways in a disease called sickle cell anemia, then it cannot carry as much oxygen. It also produces these sort of sickle cell shaped red blood cells, and they can lead to complications with blood flow through the circulatory system. So why do these occur? Why is there a change in the hemoglobin? And why does that lead to sickle cell anemia? Well, as it turns out, there are two ways how the regular allele, which is called the A allele, is potentially changed. Now, let's think back about alleles. We talked about alleles briefly, but we haven't really spent that much time with them. An allele is a genetic variant of the same gene. So you have your gene, and there are, let's say, two different variations of that particular gene. And so it still codes for the same thing. So it's hemoglobin in this case. All three alleles, all three variants are coding for hemoglobin, but the hemoglobin has slightly different characteristics. And so that is what's called an allele. If you have a change in the DNA nucleotide sequence here, then this is a different version of that. And so therefore, this is an allele, a different allele of that. This is also an allele, a different allele of that, right? So it's not that one is the normal thing and the other one's the allele. They're all different variants of each other. Okay, so you see here the A allele, the S allele, and the C allele, and they lead to these changes in these particular amino acids, lysine, valine, or glutamine, and these two are mutants that change the protein's function. So that's an example of how alleles can change the function of a certain protein. In the case of sickle cell anemia, the interesting thing is that the change does not necessarily mean only that that's a bad thing. So oftentimes a change in a gene, a mutation in a gene, can be detrimental, so can be bad, can be, potentially can obliterate the function of the gene completely. In the case of sickle cell anemia, yes, that is a problem because of the shape of the red blood cells. But it turns out that can also lead to resistance to certain to another certain disease, to malaria. So people with this particular allele, the S or the C allele, are more resistant to malaria than people with the A allele. Yes, they have problems with the blood, but they potentially have an advantage because they're not getting infected with malaria. So of course, this particular allele might only be of advantage for people who live in an area where malaria is a problem. But I'm mentioning this here because I want to be sure that we understand that alleles are variants, that they're not necessarily good or bad. They are different. And so they can change the function of the protein, and that protein can then potentially have just a slightly different function. So here's another example. There are some alleles that particularly act together with the environment. So what that means is that depending on the environment that they're in, so the individual with that particular allele, if that particular organism, in this case it's a person, is in a certain environment, then having a particular kind of allele will maybe potentially compound certain aspects of a disease. So let's take a look at this example here. Here we have an allele in this particular gene that's called alpha-1AT gene. So this particular gene, this is the chromosome. Here's the little red bar that tells you where on the chromosome that gene is located. It produces this enzyme, and that enzyme inhibits elastase. And elastase elastase has something to do with the elasticity in the lung of a person, of a human. So turns out that in the case of when the environment is smoky, when people smoke, but it could also be secondhand smoke, that reduces the activity of the alpha-1AT, and that decreases the inhibition of elastase, and that leads to increased elastin breakdown, and that can lead to emphysema. So that's a lung disease. It's not really so important that you understand this pathway of elastase and elastin and emphysema. That's not what this is about. What this is about is the fact that here's a gene and here's an environment, and there's an interaction between the environment and the gene. Under certain conditions, this particular gene is affected, in this case by cigarette smoke. Now, in addition to that, that normal gene can also be mutant. So there can be an allele, a mutant allele, and that's called the PiZ allele of this particular gene. Well, that PiZ allele also produces a condition where individuals who are homozygous, that means they have 2 copies of that, for this PiZ allele, they produce alpha-1 AT with reduced activity, leading to increased elastin breakdown in the lung. And so that can also lead to emphysema. So it turns out if you have this particular allele and the environment is conducive for this allele to be particularly detrimental, then that becomes even more problematic for the organism, for the human in this case. So the way I'm summing this up here is that allele variation leads to phenotypic variation, and that environmental conditions can interact with specific alleles of a given gene. So in other words, in this case, under the given environmental conditions, smoke, having a particular type of allele can be much worse than just being in that condition or just having that particular allele. So yes, smoking will increase your chances of emphysema, and yes, having the PiZ allele will increase your chances of having emphysema, but doing both or having both makes it even harder, makes it even worse. So there's an interaction between a particular allele and the environment. Okay, so and then there are mutations that can be advantageous. So there is a particular allele that researchers have found that are on a particular type of cell that humans have in their bloodstream. So those are called T cells. T cells are important for helping with infections. And within this membrane of the T cell is a particular surface protein, and there's a receptor. You see this receptor here through the membrane. Here's another receptor. Well, they do something, and what exactly they normally do is not relevant here. But it turns out that the virus that causes HIV also requires an interaction with these 2 surface proteins, with this co-receptor and with this surface protein. That's something that obviously the T cell didn't evolve to do, right? It's the other way around. The HIV virus, in order to infect the cell, has over evolutionary time adapted to the fact that these receptors are always on the cell, and so it has used that sort of as a way of recognizing that that is the type of cell that it wants to infect, that it is good at infecting. So that's why it says here, for HIV to invade a T cell, it must interact with the CD4 receptor and the CCR5 co-receptor. So this interaction has to occur for the invasion. But it turns out that a mutation in the T cell membrane receptor, in these receptors here, increases, or I should say decrease the likelihood of HIV infection, because it decreases the likelihood of the interaction here. So you change the proteins here, and you are no longer supporting the kind of interaction that HIV requires for entry into the cell. So that's an example of a mutation that has an advantage for the population, or for the individual that has it, and the population that might have this particular allele more frequently present. And the reason why that is the case is shown on this particular slide here, where you have the non-mutant CCR5 allele. So that is the gene, the CCR5 gene. And in this version of the gene, that allele, it has this set of amino acids here, right? From here, from the serine here to the threonine, it has these particular enzymes these particular amino acids. But in this mutant that I was mentioning, that is less likely to bind with the HIV virus, there's a deletion from here to here. So the entire red part here is deleted. It's gone. It's just mutated out, if you will. And that deletion causes a shift in the translational reading frame. And all downstream amino acids are incorrect. So what that means is that the last tyrosine here, the TAC, is still correct. But then when it comes back to this A here, you see that there are changes that are always in brackets of 3, right? These 3 are codon, this is a codon, this is a codon, this is a codon. And here, the mutation ends at the C, not at the A. And so therefore, the translation will occur from the TAC to an ATT. And there wasn't an ATT before. The next amino acid was TAA. So now it reads ATT and therefore puts in the correct amino acid for the coding sequence ATT, and that's isoleucine. And so that's why it says here the downstream amino acids are now incorrect from the normal allele. And then in addition to that, there's also a stop codon that will terminate the translation of this protein. So here you see how a severe mutation, so a severe deletion of a piece of DNA that has both the effect of a change in the reading frame and a premature stop codon, actually can have an advantageous, can be an advantageous event for this individual. Okay, so we've looked at a number of examples, and in each case, there was a mutation in the DNA that leads to a change, and to the formation of 2 different alleles. So the question is, how do you detect that? How do you know that a person or any organism that has DNA has one or the other allele? And when you're doing this kind of analysis where you're asking which allele does it have, that's called genotyping. So the genotype is the information about what allele a certain organism has. And in order to do the genotyping, there are a lot of biotechnological tools that can be used. And I want to show you a few. This actually comes from a different chapter that we have jumped over so far, but we will come back to it. The reason why we've jumped over so far is because it fits better the way that we have it here with the lab. And the reason why we have it, why we have in the lab a certain sequence of things has to do with your project, with your long, semester-long project. So bear with me a little bit while I tell you about these biotechnological tools, and then we'll go into more detail about these later on when we get back to chapter 12. So when you're looking for different alleles, what you can do is you can extract DNA. You take the cells, you extract the DNA from the cells, and then you use a technique that's called gel electrophoresis. So this is a illustration of gel electrophoresis. And I want to show you what gel electrophoresis does. So basically, what you do is you make a gel. This is the gel. And the thing that it's in here is a box. It's a gel box. You will actually later on in the semester see one. You'll run a gel. You'll do this kind of analysis. The gel box has a buffer, a liquid, in which the gel is bathed. And it has electricity that's fed into these electrodes here. Here's the negative pole, here's the positive pole. And this gel is sort of like Jell-O, and it actually is basically made like Jell-O. So you take a powder, you pour boiling water on top of it, you stir it, or you do this in the microwave, and then you pour it into a tray, it's called. It's just when you make Jell-O, you pour it into a bowl. Here you pour it into a little tray so that it's nice and flat and square. And into the tray you put it's called a comb. It's basically an insert that will leave little holes that you will have in the gel when you remove the comb. So that's sort of like if you make Jell-O and you put a spoon in there and then it solidifies and then you take the spoon out, you rip a hole into the gel. that's what's happening here as well. So you make these little holes by putting a comb in there and then taking it out after it solidifies. And then you can insert the DNA samples into these little holes. They're called welds at that point. So they're on the edge of the gel, and you insert the DNA that you extracted, that's in a liquid, in water, in a buffer. And then you apply this voltage. You put buffer in there as well so that the liquid can transmit the electricity. And then the positive pole here will attract the negative charges in the DNA. So you have DNA here, and you have a attraction force over here. And so the DNA fragments now move towards the positive pole, and it says here according to their size. So if you have small pieces, they will go pretty fast. If you have long pieces, they will take a while. I always liken it to that picture, like if you think about, you have a bunch of people and you chain them to each other, handcuff them to each other, 2 people, 3 people, 5 people, 20 people to each other. And then you send them through a forest and you say, run and make it to the forest, through the forest as fast as possible. Well, if you have a group of 2 that are chained together or a group of 20 that are chained together, the one that's most likely to get out faster is the one with 2, because they can wiggle through the trees much faster than a group of 20 could, because they always have to sort of, all go around the tree in the same place, in the same order, and fold up a little bit so that they can get around that obstacle. And that's really what's happening in the gel as well, where the obstacle is not trees, but molecules of that the gelling agent. So in case of Jello, it's gelatin. In this case here, it's agarose. It's a type of sugar, but it's essentially a gelling agent. So it's a molecule that these DNA molecules would have to wiggle their way through, and so therefore, the shorter they are, the faster they will get through this gel. So to recap, you put the DNA in here, you put buffer in here, you apply a charge, and the charge will pull the negatively charged DNA through the gel, smallest pieces first. These are then called bands. These individual pieces that you see are all pieces of DNA. It has to be stained, so you stain it in a certain way, and then they're called bands. And in this picture shows you what it actually looks like. Right? This is a drawing. This is a picture of what a electrophoresis gel actually looks like, with pieces of DNA, discrete pieces of DNA that were stained with a fluorescent dye. So what's important here is that for gel electrophoresis, DNA is isolated, then it's often selectively amplified so that there's enough that you can see it. And that amplification reaction is called a polymerase chain reaction. We'll talk about that later. You'll actually do it in lab. But for right now, all you need to know is that it amplifies, it makes lots and lots of copies of the same pieces of DNA that you put in to that reaction. And then once you have that, you size select these pieces on a gel. Okay, so if you know that you can size select DNA pieces on a gel, then you have what you need to understand this next slide. So we were talking about variations. We were looking at the kinds of different alleles that arise from mutations. I said that we wanted to find a way to genotype an organism to see whether they have one allele or the other allele. And then I said that these biotechnological tools can be used for that. And I want to show you how they are used and what they're called. So if you genotype something, or you have genotype identification by allele lengths, you can use a technique that's called variable number of tandem repeats analysis, VNTR analysis. So what is a VNTR? What's a variable number of tandem repeats? So this is illustrated here on the side. So we have 5 different alleles. Right? So far we've looked at cases where 3 alleles in the case of the hemoglobin gene at the very beginning, where we had the A allele and the S and the C allele. Here we have 5 alleles, and this is called A1 through 5. And this is our particular gene here. And these 2 pieces, they're called PCR primers. We'll talk about that later. Essentially, they are used for amplifying this piece of DNA. So if you only have one copy of it or a few copies of it, that's not usually enough to see on a agarose electrophoresis gel. And so that's why they get amplified. They get amplified by this technique that's called polymerase chain reaction that uses molecules here and here that bind specifically to the ends that you want to amplify. And then there's a way how you can you add some enzymes and it will make multiple new copies of each of these two strands. We'll go over that in much more detail later. For right now, all you need to know is that you can amplify these pieces. But we wanted to look at the alleles and we wanted to see what is different between them and why they have variable numbers of tandem repeats. So you see the tandem repeat here that always is TAG, 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 TAG. So it's repeating the TAG motif, and it's doing that, you know, a tandem is a bike that has the same thing twice next to each other. Right? So that's basically what's happening here too. This piece is twice right next to each other. In fact, it's even 3 times, 4 times, 5 times. Many, many times it's repeated. And so a tandem repeat can be either repeated just a few number of times or a large number of times. And the number of times that this motif is repeated is what varies between these alleles. So here you see that this allele only has 1 copy of this repeat. This allele has 2 copies of the repeat. This has 3 copies of the repeat, and so forth. And so 2 organisms might differ in the alleles that they have. They might have an allele number 1, and they might have an allele number 2, one that they got from their mother, one that they got from their father. So then they have a variable number of tandem repeats of 1 on one chromosome and of 2 on the other chromosome. But it's also possible that they inherited a chromosome that has an allele with just 1 copy or one with 5 copies of this particular repeat. So now we are extracting these DNA molecules and we're running them on the gel. And I said that the shortest pieces will run the fastest through the gel. So this guy over here, A1, will actually run the fastest. And then the next one, this will be a little bit longer because it has the extra copy of the tandem repeat. And so that'll take a little bit longer to wiggle itself through the gel. And then the one that takes the longest is the one with the 5 copies. So you can see here how they stack up when you run them in a gel, when you load them up here into these wells, these holes in the gel, and then you apply the positive charge down here and you suck them through the gel. And then you wait, you stop after a while, you look, you visualize the gel, and you see these bands. You see, the band is over here, there's a band over here, there's a band over here. This is a preparation of DNA that had exactly 2 copies, copies from both chromosomes with the allele A3. Okay, so on this particular gel on the left, this is just showing the position where each of these pieces of DNA would be localized after running them through the gel. Now, if you have a combination, if you don't have just A1 and A1, so this from the mother, this from the father, if you have one from the mother that is A1 and let's say one from the father that's A2, you will now have 2 copies in there, right? So you will have this one that runs the same size or the same speed as the A1, and then this one that runs like the same speed of the A2. So this would be an organism that has 2 different types of alleles, one on each of their homologous chromosomes. This would be a case where you have also 2 homologous chromosomes, but they have the same allele. So they have the same type of variant on both chromosomes, and so that's why you see only 1, because they're on top of each other, right? From mom and from dad, they look the same, and so therefore you only get 1 band. But if from mom and from dad looks different, then you get 2 bands here. But you would never get 3 or 4 bands in this case, because each allele exactly shows one band. So now you see all the different combinations, right? You see that maybe 2 and 3 would look like this, 3 and 4 would look like that, 4 and 5 would look like that. This is called a fingerprint, a genetic fingerprint. Each of the different genotypes yields a unique pattern of bands in the gel. So when you hear the term genetic fingerprinting, what you can do is you can create a pattern that is totally specific to a person, and it could be either this pattern or it could be that pattern. And so if you imagine isolating DNA from one person and from another person, or from a tissue in another tissue, or from a blood stain at a crime scene and a person that might be the suspect, you can see whether they have the same patterns, the same genetic fingerprint. And so if you find this in your crime scene and you find this fingerprint in the suspect, can't be the same person, right? That would exclude that person from, at least from being the source of this particular DNA sample. So what I write down here for you to remember is that for VNTR analysis, a gene with multiple alleles in the population is amplified by PCR. That's what you see here with these primers. Again, we'll talk more about PCR later in an upcoming lecture. Longer alleles take longer to travel through the gel. So these are the longer alleles. They take longer. They have only run from here to here. These have run this far much faster. And then diploid organisms like humans, we have one chromosome from mom, one from dad. They have 2 copies of each gene, one on the original homolog from the mother and one on the homolog from the father. And if you have 2 copies of the same allele, just like in this case, 2 copies of the same allele, both A1, then that is called homozygosity. And if you have 2 different ones, then that's called heterozygosity. So this is a heterozygote. This is a heterozygote. This is a heterozygote. And these are all samples from homozygotes, right? Because they have exactly the same allele on both chromosomes. Okay, so that is one way how you can use biotechnology to analyze variants between organisms. So there's another way to do it, and that is called, well, it has a number of names actually. Let's just call it SNP analysis for right now, and then I'll show you another name for it in a second. So SNP analysis stands for single nucleotide polymorphism analysis. What is a single nucleotide polymorphism? Well, first of all, what's a polymorphism? A polymorphism, poly means many, and morph means variations, exactly. So many variations. A polymorphism, let's say between 2 organisms, let's say butterflies. Sometimes there is maybe a black and a white type of this butterfly. And so they're called morphs. They're called the white morph and the black morph. Otherwise, they're the same species, but one's white, one's black. And it also happens in tigers, right? White tigers and regular tigers. Different morphs. So when you have multiple morphs, ways of appearing, then you have a polymorphism. So that can also happen in the DNA. You can have multiple appearances, that means different types of nucleotides in this case, for a particular trait. And let's say that trait is this particular position on the DNA, on the chromosome. Okay, so what do I mean specifically? I mean, here you have a GAG sequence that's over right here. And here you have a GTG. So that is a polymorphism because it looks different between the A allele and the S allele. And the reason for why it looks different is because there's an A here and there's a T here, right? So when we have our orange and our white tigers, that's easy to see by eye. This is not so easy to see by eye, but it's the same idea. There are multiple ways how this particular trait can appear. So it can either appear as an A or it can appear as a T. Okay, so once you know that, you can use something that is called a restriction enzyme. So a restriction enzyme is an enzyme that originally comes from bacteria, and bacteria use restriction enzymes to cut DNA into pieces from invading viruses. Bacteria also get infected by viruses, And they have these restriction enzymes as a way to defend themselves. So DNA gets inserted into the bacterium. They take a restriction enzyme, which is an enzyme that cuts DNA, and it cuts it at a very specific sequence. It has to cut it at a sequence that these bacteria don't have themselves, otherwise that enzyme would cut their own DNA. So it cuts at a sequence that they never have. And that's where biotechnology originally got its restriction enzymes from. So there's a whole slew of different restriction enzymes that recognize different sites, different DNA sequences. What this says here, a restriction enzyme cleaves both DNA samples at all of the sites it recognizes. And so a specific restriction enzyme only recognizes a very specific site. So let's say we have an enzyme that recognizes GAG, but it does not recognize GTG. So this enzyme now binds here and it cuts the DNA because it recognizes this sequence in this context and it cuts it. And so the reason originally for why it does that is because this might be the sequence that's found in a virus, and this particular bacterial restriction enzyme cuts that viral DNA. Well, if we isolate DNA from an organism with allele A, and we use this particular enzyme that we have isolated from bacteria, you can now buy these in a biotechnology store, essentially. So they come in a little tube, so there's no bacteria involved. But if we have DNA isolated from an organism that has allele A with this sequence, and we're now adding that restriction enzyme, it will cut right here, right? It cuts here. Now you have two pieces. You have this piece and you have that piece. If that individual, or if a different individual has the allele S, that is has the polymorphism here, where the T is substituted for the A, then that enzyme can no longer cut this piece. It just cannot do it because the T is in the way. That enzyme can only cut when there's an A. Remember that enzymes are really, really finicky, really specific with what they do, where they bind, and how they act. So this restriction enzyme will not cut here because of the T. And so therefore, that piece is uncut and stays in one long bit. So that means that when the restriction enzyme cleaves both DNA samples at all of the restriction site it recognizes, a restriction site is the site that the restriction enzyme recognizes, then it produces fragments of different lengths. Here we have two fragments of a small and a larger length, and here we have one fragment of a very large length. And then following electrophoresis, the gels show the position of the fragments to which the probe hybridizes, or where the DNA runs. So what it means here is that this piece, when you run this on a gel, will run relatively high up in the gel because it's a large piece of DNA. These two pieces will run much lower. So now, your book talks about a probe and it talks about hybridization. That's yet another technique. Forget that for right now. Just think about what it would look like on the gel when you run a large piece versus two small pieces. Well, it would look just like this, where you have the gels differing from each other with a fingerprint, if you will, of having two pieces here and one piece here. Okay, so there's one more thing I want to mention here, and that's that this particular example from your book actually has multiple restriction sites on each of these alleles. So it has a piece of DNA. It has 3 restriction sites here. It just says, okay, this is an example where you have 3 restriction sites. This enzyme will cut here, and will cut here, and will cut here. And in this case, it will cut here, and it will cut here, but it will not cut here, right? So that is what's shown here, why there are 2 red bars here and 3 red bars here. What you need to remember is only that the enzymes will specifically cut, and they will then produce these very specific restriction fragments. So that's what they're called. This is a restriction fragment. So I have the arrows pointing here. So these are 2 smaller restriction fragments. This is 1 larger restriction fragment. Okay, so if you look at this example here, where we are detecting single nucleotide polymorphisms from different organisms, from different individuals, and we are using that for fingerprinting. What I want to point out is how this particular analysis can be used to determine whether an organism is homozygous or heterozygous. So this example here produces is an organism that has two alleles of the same kind, the A allele and the A allele. And then this organism has the S allele of both kinds, both from mom and from dad. And this organism has an S allele and an A allele. So this is the A allele that's over here. That's the S allele over here. Mom, dad, mom, dad, mom, dad, right? So if you are isolating the DNA from both individually, and then you cut the DNA from both individually, and you run it on a gel together in the same well, in the same lane here, you would see that that enzyme will cut, of course, exactly the same way because it's the same allele. Doesn't matter which individual it came from. And so you will have one large band and one short band. And then for the S allele, if it's the same, then you'll get one large band will run on top of each other, right? Moms and dads will run exactly the same size, and so they will run on top of each other. In both cases, we'll call this homozygous. This is the homozygous pattern for the A allele. This is the homozygous pattern for the S allele. If you have a heterozygous organism, so one that received a chromosome from the mother, let's say, that's the A allele, and one from the father, that's the S allele, then that will give you a combined pattern, right? This will be the pattern that you get from here. The large piece will be this, and the larger but smaller than this, and the very small piece will run down here, and that will come from this allele. And that will be a case of heterozygosity. So here we have a heterozygous organism with the genotype AS. Here we have a homozygous organism with the genotype SS, and here we have a homozygous organism with the genotype AA. And this kind of analysis, when you are using restriction fragments, when you're cutting DNA with restriction enzymes, and then you're analyzing their variable restriction fragment lengths, that is called a restriction fragment length polymorphism analysis, or for short, RFLP analysis. So this is RFLP analysis. Again, just like before, it uses a polymorphism, a difference, can be a SNP, to determine different alleles in different individuals. All right, so let's take a look at an example. SNPs can be detected the way that we just discussed, and they can be associated with a phenotype. They can be associated with a phenotype directly by changing a gene, or they can also be, let's say, indirectly be associated with a phenotype by having something to do with the transcription or the way that the gene functions that's not exactly in the gene. So maybe upstream where the regulatory region is, where it determines whether or not transcription occurs or not, that can also affect the function of a certain gene. So here's an example where you have a SNP, where you have a single nucleotide polymorphism that is for blue eyes or for brown eyes. So we have the GC allele with the TA allele. So in this case, we have people with the CG allele are much more likely to have blue eyes than those with the TA allele. Okay. Another way of variation that can occur between two different individuals can be not through a SNP, not through a mutation, like a truncation mutation or a deletion mutation, but by something that's called a copy number variation mutation. So what's a copy number variation mutation? So a copy number is basically the number of copies of a gene that exists on a chromosome. So see here, this chromosome has one copy of this particular region. Let's say it's just one gene. Could be longer than a gene, can be, several thousand nucleotides long, but let's just say it's one gene. through a variety of mistakes that happen all the time, and they may not always be repaired, but sometimes might be, changes can occur when the chromosome is duplicated. So when that happens, it's possible that a chromosome piece can be multiplied, can be duplicated. And that is when you have a transition from one copy to two copies of this particular gene or region. So in that case, we have a copy number variation between this chromosome and this chromosome of 1 versus 2, or no copy at all. So copy number variation can be important. It can certainly be important when the gene is deleted, but it can also be important when the enzyme that's produced, or the product that's produced, is simply more active because there's more of it there. More copies of it might mean more transcription of this particular gene, having more of an impact on the organism. That doesn't always happen, but it can happen. And here's an example for when it can happen. So some genes that are duplicated in some populations are amylase genes. Those are starch digesting genes. And on this slide here, you see the proportion of chromosomes, so the number, the percentage, if you will, if this was 50.5 proportion is 50%. The number of copies of this particular gene, that's the amylase, the starch digesting enzyme, on chromosome 1. So the number of copies that are found on chromosome 1 and how frequently that happens in populations. So about half the time, a little bit more than half the time, it turns out that people who have a low starch diet, or populations that don't eat much, potatoes and wheat, things like that, they will have, with a low starch diet, they will have up to 3 copies of, or 60% will have 3 copies of this AMY1 chromosome. And people with a high starch diet will only have 0.2. So in societies with low starch diets, the average AMY1 copy number is chromosome 1. On chromosome 1 is about 2.5, where it's without, it's 3.5. And then in proportion of the population with a high starch diet, there are some people that have as many as 5, 6, or 7 copies of this particular gene. So in high starch diets, it's much more frequent that you have more copies of AMY1, and in low starch diet, it's much less frequent, right? There's hardly even maybe 4% of people that have 4 copies. None of them have more. Much more frequently is to have a fewer number of these copies. So copy number variation can have an effect on how well a certain process works, simply by the number of molecules that are being produced. Okay, so yet another way how you can have variation, genetic variation, is by having a change in the number of whole chromosomes. So far we've talked about mutations, changes in the genes. Then we've talked about duplicating or losing entire chunks. So that was copy number variations. And now we're talking about losing or having extra entire chromosomes. So that would be a huge change in the amount of DNA for a particular kind that a certain organism has. And that can happen during meiosis. So remember meiosis, we have metaphase 1 where the homologs pair and then get separated, and then in metaphase 2 the sisters get separated, and we have 1 copy of each of the chromosomes in the gametes that are produced. Well, that's what normally happens. But in some cases, there's a process that's called non-disjunction. This is something that happens when something goes awry. So think about meiosis here, the pairing of the homologs. Well, when the spindle fibers attach here, maybe one of these spindle fibers attaches over here rather than this spindle fiber attaching at the centromere. That can happen. Usually shouldn't happen. There is a checkpoint that makes sure that that is not the case. But when that checkpoint somehow is not working, then there is a possibility that both chromosomes are moved to one pole. And in that case, you have no chromosome here and you have two chromosomes here. And that means that in the next generation, in the gametes, you either have two copies or no copy. So non-disjunction results in gametes with an extra chromosome and gametes that are missing a chromosome. So I say here non-disjunction means that homologous chromosomes fail to divide normally during meiosis. Well, there's another way how this could happen. This meaning a change in the total number of chromosomes in the gametes, and that is that in the second division you may have non-disjunction. So you may have perfect disjunction here. So each of the two homologs go to the next daughter cell. But then here you have a lack of non-disjunction. In this example, where both of the sister chromatids separate from each other, but both move to the same pole. So now you have an extra chromosome here and a missing chromosome here. So that means gametes with extra or missing chromosomes lead to chromosome variation after fertilization, right? So if this one now was to be fertilized with a typical cell that has one chromosome, then it would have only one, not two. It should normally have one, it should bring one, and then the one that it's fusing with should have one, so there should be two. But if it's missing one, then there's only a total of one. Likewise, if this one were to fuse with a typical gamete that has one chromosome, then it would have a total of 3 chromosomes, not 2, which is the norm. So that can happen, and that happens in animals and in plants and also in humans. And your book has this figure that shows the incidence of extra or missing chromosomes in human live births. So what that means is that in many cases where changes occur with the total number of chromosomes, that is so detrimental that in most cases the birth does not result in a live birth, so that fetus is aborted before the baby is born. But there are some differences that chromosomal differences that can lead to live births, and this is what's shown here. So in the cases where extra missing chromosomes do not result in spontaneous abortion, but in a live birth, then those live births are most likely to have a chromosomal abnormality of an extra 21, an extra chromosome number 21. That's what this pie chart shows here. So out of 10,000 human live births, there are about 33, statistically speaking, 33 births where the individual has either extra or missing chromosomes. And most of those, they have 47 chromosomes, and that means that in this case, it means that they have an extra chromosome number 21. And that is also known as Down syndrome. There are other cases where you have, for example, only a total of 45 chromosomes, so there's one missing, and that's one that has an X chromosome but not a Y chromosome. And then there's that's called the Turner syndrome, and then the Klinefelter syndrome is the one where there's an extra chromosome, so not 46 but 47, and that has an extra X and a Y chromosome. And then there's also these possibilities, an extra 18 of the somatic chromosomes, an extra 13 of the somatic chromosomes, triple X individuals or XYY individuals. So when you isolate cells and you look at the chromosomes under a microscope, we looked at these kinds of graphs before, or they're not really a graph, it's called a karyogram. So you isolate the chromosomes. These are real chromosomes, these are not drawings. And then you cut them out, and with a in the past with scissors, but you can do this now with a computer, and then you align them next to each other by size. So these are the two chromosomes number 1, these are the two chromosomes number 2 from an organism. They get numbered, they also get put in certain groups, And then you see the sex chromosome. So in this case, this is a female XX. And here you can see an example of a trisomy 21. So tri means 3, somy means the body. Autosome is often referred to as a chromosome that's part of the non-sex chromosomes, so an autosome. And then you would say a trisomy means it has 3 bodies. It has 3 number 21 chromosomes, and that is what it would look like in one of these karyograms. You can spend some time on your own looking at this information here. It's quite interesting to see exactly what the types of chromosomal variations that there are, how many of these can lead to life-born, and how many can occur which are spontaneously aborted. And then here's an example of sex chromosome variations. So when we are looking at the Klinefelter symptoms or the Turner symptoms, these are females. They have an X chromosome and no Y chromosome. And there's a few things that are through variants that look different in Turner syndrome individuals, as well as in Klinefelter syndrome individuals. Okay, so I want to finish this by telling you that there's also a possibility that you can not only increase the number of whole chromosomes, but you can also increase the number of whole chromosome sets, and that is called polyploidy. So in the case of polyploidy, you're getting entire chromosome set duplications. So here's an example where this happens, but they're only showing you one chromosome. And so it looks very similar to the examples that we had before. But take a look at what happens here. So we have, in this case, an example where this goes through no cell division. We get duplication, but we don't get cell division. We have 2 chromosomes in the end in diploid gametes. And we have a regular haploid gamete here. So this is the normal haploid gamete. Here is one where cell division didn't happen, but DNA replication occurred. So imagine now that you have a diploid gamete being fertilized by a haploid gamete. Well, then you get a triploid fertilized egg. That's very similar to what happens if you have a case where you have an extra chromosome, let's say chromosome number 21 here, and this is just one number chromosome 21, and this is 2 number 21 chromosomes, then you would have a trisomy. What this is trying to show you is an example where all the chromosomes, and they didn't fit all the chromosomes in this picture, but just one example, where the entire chromosome set is triplicated. That is called triplication, triploid organisms, and they can then produce diploid or haploid gametes. So a triploid organism is very unstable. Doesn't happen very frequently, certainly not in humans. Out in nature, it's relatively infrequent as well. Actually does happen in some cases. Those are very odd organisms. And then here's an example of a tetraploid formation. So here, where we have DNA replication occurring, but no cell division. So we have DNA replication in S phase, and then we go through mitosis, but we omit cell division. This is a mistake, right? This should not happen. But if it happens, then it leads to a tetraploid cell. And tetraploid organisms would produce diploid gametes, right? So they will then duplicate these and then maybe go through regular meiosis, and they will always produce diploid gametes. So tetraploids are relatively stable among the plants, not among humans. There are quite a few animals as well, frogs and fishes and so forth, that are polyploid. But if we look at this table here that I showed you earlier, see, this can happen in humans. This is for humans. This is a triploid organism that occurs, but they are almost always spontaneously aborted. So there's very, very few examples where a child was born with triploidy or tetraploidy. Okay, so let's finish it up with a summary. We talked about changes in the DNA sequence, and we said that that leads to mutations. We said that mutations can be detrimental, they can be advantageous or neutral. Variations in a gene are called alleles. Alleles can be detected by biotechnological tools that we discussed through gel electrophoresis. I talked a little bit about polymerase chain reaction, but I said I will come back to that in a later lecture. Variable number of tandem repeat analysis, the VNTR analysis, and the RFLP analysis, so that you should look at. We talked about the fact that problems during meiosis or mitosis can lead to a change in the number of individual chromosomes. So the state of having more or fewer chromosomes per cell than typical, that is referred to as aneuploidy. I didn't mention that term earlier on, but we did talk about this, and I wanted to mention this term of aneuploidy to you here. In humans, aneuploidy is often lethal, but some variants are viable, and we took a look at some of those. We said that the Klinefelter syndrome, for example, the Turner syndrome, Down syndrome are all examples of viability of aneuploidy in humans. And then we talked about polyploidy. I said that that arises from whole genome duplication, not just single chromosomes. And we said that in humans that is very, very uncommon, but in plants and some animals, polyploidy is actually quite common. All right, and that is it for today. I will see you then in class. Bye-bye.